All right, let's walk through the major features of the cell annotation platform. So this is the home page. You'll see on the upper right, we have an invitation for scientists to register and log into the site. Two reasons for this. First is for user roles and user management of data. So we need users to log in such that they can access only the data sets that they have access to. Um, you'll see that we have this organizational structure that we call a workspace, whereby you can add collaborators to either view your cell annotations and your data sets or edit them. This is very similar to, let's say, a Google document. When you open a Google document, only you have access to make changes to this Google document unless you add collaborators. And then you can select their user roles as viewer or editor. A viewer can only view the Google document and an editor can make changes. And the same principle applies to how we've designed CAP, which I'll walk through. Second reason why we ask users to log in and register relates to this idea of public reviews of the cell annotations, the feedback that one scientist can give to the cell annotations of another scientist. And so we need users to log in and register to do this, but we also need the scientists to be publicly identifiable. And this is why we ask them to create a profile upon registering to the site. So I've already created a profile, but I can log in. This is very standard. One adds a email address and a password. This is my publicly accessible profile. It has things like my name, my ORCID ID, my email address, location, institution. It has, I can quickly edit this if I wish. And these are what I was describing earlier. These are only accessible to me. You could think of these collections of data sets as what we call workspaces. These are these collections of data sets that only I have access to unless I invite collaborators to work on these data sets. And so if I look at, let's say, this, you'll see there's two data sets in this workspace, and there are others who have access to this. You can see their user roles to the right. This is an owner, an admin, editor, and there's no viewer here. And the idea is that users should provide a subject line or a title for this collection of data sets and some details which we haven't provided here yet in this example. Why do we do this? For a lot of reasons. In terms of papers, usually it's not, a paper does not correspond to one data set. Sometimes usually there's multiple data sets associated with a given paper. And this is why we've organized this idea of collections of data sets, or what we call workspaces. You can see everyone here, they have edit permissions to make changes to these cell annotations. So that's something only accessible to me. As soon as I publish it, it becomes what we call a publication. I can go back and I won't use my buttons on my web browser, but use the buttons on the site. So that is these are private data sets only I can work with and my collaborators. This is my profile. Now we can see the data sets that are published on the site. There's two ways to search for these. We have a table for the data sets themselves. You can see we have columns of data set, organism, sequence, tissue, disease assay, number of cells. We could also check for, let's say, the publication name and the authors. And there's standard UI widgets here to sort by alphabetical order or descending order. Or I can search, what should I search? Let's say retina. I see the word retina. So I search for retina. I should be able to access only those data sets. I can also make queries here using the facets of certain tissues that exist. And so if I click on heart, I'll see the data set that's associated with the heart. So go back to default view. That's one way to look for the data that's been ingested on CAP. Another way is with the cell labels, and this is really the heart of the matter, the cell annotations, how scientists have defined these biological entities. Not surprisingly, at the top, we have an immune cell. I mean, immune cells happen in most tissues in the human body. And so they're fairly popular across, as you can see here, about many different contexts. And we'll see how the scientist has associated these within a common ontology term, which is associated with biomedical databases. I can click a look to see which, this is just querying the database to see how this B cell is defined. But here you can see that the scientist has provided synonyms for this entity, marker genes, the organism tissue. If need be, I could see which scientist this was, the number of cells, canonical marker genes, the category term for this cell, etc. And it's fairly straightforward to search for these. If I click on this entity, I'll go to the data set, and the context of how precisely this B cell was defined within this data set, you'll see that there's two sets of cell annotations here. I can see the connections between these two sets of cell annotations. So for instance, an epithelial cell in this set of cell annotations corresponds to fairly fine-grained cell annotations in the second set of cell annotations. So to make, there's two sets of cell annotations here. One is major cell type and the other is author cell type. Perhaps unsurprisingly, author cell type is a bit more detailed, which is why we see these connections here, whereby mesenchymal is a very broad term. When I click on the author cell type, you'll see these very detailed cell labels for a mesenchymal cell. And if we go to molecular data, we could actually look to see how these cell annotations have been, how they really interact with the molecular data itself, which is really the important thing here. So as opposed to simply checking the context of how these cell annotations were defined, I can see, as I said, these are going to be very broad cell types, these cell labels here, and this is, of course, how this has been defined. Major cell type, you can see the author cell types. If I color the embedding by author cell type, I'll see that these are more refined cell types of these broader cell types we were looking at previously. Here you can see differential expression results are being propagated at the heat map. This is slowly loading. This is gene expression, and so that is basically a quick, there we go. That's a quick view of the molecular data. Let's go back to, well, we also have documentation. You'll see there's documentation that explores, that introduces users how to work with everything on the site. Let's take another look at a data set here. So if I look, we'll see an atlas of cells in the human tonsil. Let's have a click on this. This was a fairly new paper that came out recently. If I wanted to see the publication, I would click on this. I'll see pictures of the scientists. I happen to know that Ramon is a postdoc at Weill Cornell Medicine, and then Sergio is a colleague at Massachusetts General Hospital. This is the summary of the publication, which gives some context of what this was all about. You'll see there's maybe close to 400,000 cells that were sampled by 17 different healthy donors of across various age groups. And if we want to look at the data to see actually precisely what this means, I'll be able to actually see these are the total number of cells, age group for some of these cells, the tissue that was used during the sampling, sex of the patients, and so forth. And there we will have the name of the cell annotations. Each one of these cell annotations has further context, which we can click on and look at. And we can look at the molecular data to see really what do these cell annotations mean in terms of the data. And so on the left, I'll see the number of cells associated with the cell annotations. I can take, again, look at the context of these cell annotations. Here, B naive was associated with a B naive cell, the ontology term in these biomedical databases. These are synonyms that the authors provided. Category, which is sort of the nearest broadest term, the nearest broader term to a naive B cell. If you think of cell types, there's various ways that you can annotate them in terms of precision. And so sort of the next step up in terms of a more abstract biological entity is what we call a category. And so B cell, the subcategory of a B cell is the naive B cell. It's a bit more a precise term in this context. You can see the marker genes that were used to define these B cells, canonical marker genes, the rationale, and DUI associated with, DUI, which is associated with some scientific reasoning. There's a publication here, this DUI, that explains why it is that these naive B cells were annotated in the way that they were. We see the heat map here, which shows gene expression values for each one of these these sets of cells. These three genes were used to define B naive cells. These three genes were used to define B memory cells. I can see some common marker genes that are coming up for defining these immunology cells, these immune cells. And if need be, if I don't know what these genes are, I can basically just hover over, and we have requests to standard databases which give brief summaries of these genes and more information if need be in the hyperlink. So that is the heat map. We can also see gene expression here. So if I want to see, in terms of all the cells, what is the gene expression of this common gene, which is, let's say, the immunoglobulin kappa constant. This should be, in principle, associated with, let's say, B naive cells. And this is coloring the, it's also probably, maybe does make a bit more sense. It's associated with these platelet cells. So this is gene expression. I can search for genes if need be. Let's say genes that would probably not be very relevant, but HBB comes to mind for hemoglobin. So some of these immune cells, these are blood cells, and so I should be able to find where are red blood cells by looking at HBB, a gene which creates hemoglobin. Yep, hemoglobin subunit beta. There we are. So this is the embedding. I can select individual cell types to say, let's say, how are B naive cells defined, looking at all the differentially expressed genes for B naive cells. This is what I see in the heat map. I can expand this heat map, both expand it of looking at these specific genes, which are used to define the heat map, and by expanding the heat map in this context to look at the genes in a zoomed in version of the heat map. And these are the genes that were used to define the subset of cell types. I can close this heat map. And these interactive calculations of differential expression often take minutes, if not hours. We've tried to speed this up so it can take seconds on the web browser. Here, not only is there ability to zoom in to the embedding, or to, let's say, only select certain cells using a selection tool, which will work through the my mouse was a bit sticky there. See, I've selected 37, well, close to 40,000 cells here, and I can do things like differential expression of these cells against all other cells, or maybe just another subgroup of cells. In this case, I'll do another subgroup of cells. Let's say these cycling T cells. I'm interested to see what defines these cells in terms of differentially expressed genes with, I could use, let's say, cycling T cells. I should just be able to click on this using the correct selector. So what we'll do is take these cells and compare them against these cells to see which genes are more expressed or less expressed, and that's what we call differentially expressed. And if I calculate differential expression, this will again take maybe a couple of seconds. You'll see there's calculations that are taking place on the back end, and this will be propagated to the front end. Currently, you can see on the heat map where it is that I've made these selections. These are the cell types that I'm using to actually do this calculation. And there you can see these are the genes that were differentially expressed in terms of these selection of cell types, which include B memory cells and subtype of B memory cells versus cycling T cells. Here are some of these genes are used to, they're associated with the immune system, and they could be used to differentiate the selection of cells versus, let's say, cycling T cells. If need be, I can go back to see how precisely it is that cycling T cells are defined. So if I look at cycling T cells, and I calculate differential expression against all other cells, again, to emphasize these interactive calculations, normally via R or Python, they could take anywhere from half an hour, depends on the number of cells. In our case, we've tried to make this as parallelized as possible. And so this will take another few seconds to actually propagate this to the heat map, but these are the genes that are used to distinguish cycling T cell versus other cells that we have here. And there we are. And then scientists will be able to see these are the genes that were used and what their expression patterns are. So that is the way that users could actively look at the molecular data to do their cell annotations. What else do we have? The ability to build custom heat maps. I can look at, let's say, specific genes. I'm building these heat maps. And that will then propagate the heat map to show these specific genes, where they expressed in this embedding, and what are their identities. I can then also just do the same calculation of checking where there's gene expression for the specific gene. This specific gene being immunoglobulin number constant 3. And again, it's mostly around these platelet cells. So, with these the ability to these interactive calculations with the molecular data itself is one way that scientists will have the ability to see insights into what is the identity of the cells, and give them some hints about what the molecular data shows in terms of cell types. So